Cash. Hey guys, just see some prop the microphone, so let's see if it works. Cash. Hey guys, just see. So we go on to read the second part of the book. So yesterday, just keep in mind that since I'm testing this kind of live, so you can interact with me. I'm reading the book, The Music of Life. It's a project that I have decided to create, as I have explained the first part, because I love audiobooks. I like to listen to audiobook when I'm going to a walk. So that's the reason why I decided to make this audiobook, and I decided now to make an experiment, like make it live. So the chapter one, the CD of life, the genome. They are all in the same boat, the genes. Myth. For humans, at least, to live is to experience. How can you understand this? One thing is clear: experience is grounded in matter. The connection is there for us to draw, but drawing is quite a complicated task. And alas, complexity is unfortunate, so you are inclined to ignore it. That tends to happen, for instance, when you try to tease out the linkage between human experience and material reality. We say, it's pretty simple, really, but it isn't. Introducing the assignments. Consider an example. Before writing this page, I relaxed by listening to, for the first time for a long time, to one of my favorite pieces of music, the piano trio in E flat major by Schubert. I put the CD into my player and lay down on the sofa. As the music entered the slow movement, I cried. The emotional effect of this piece of music, which I first heard live in a concert, is always very strong. We must all have our favorite piece that has the effect on us. The effect does not always depend on the music itself. It can also depend on the context, the people you were with, and the significance of the event in our lives. So, what caused me to cry? Imagine some space traveler watching this scene. They are creatures from a world in which silicon replaced carbon. So let's call them Simons. They have some of the characteristics associated in science fiction with androids. They note the crying. They record the sound waves in the room. As scientists, they trace a sequence of cause and effect back through the loudspeaker, the amplifier, the laser disc reader, right down to the CD itself. One of them does a Simon version of Eureka. I found it, he says, as he explained to his colleagues that the whole effect is caused by some highly specific digital information in the city. Another of the Silmans is nevertheless skeptical. How, he says, could just a bunch of numbers have this effect? The discoverer counters the skepticism by pointing out that this is the lowest level of the chain of cause and effect. Without the digital information, there would be no music, no emotion. Moreover, if you play around with that information, mutate it, as it were, by playing too fast or too slow or playing backward, transposing section or even transposing bit from our CD, then the person in the room no longer cries. In fact, he may angrily turn the machine off and even throw the disc away. There is an inevitable and mechanical chain of cause and effect here. An experiment the Siemens might do would reinforce the one-way nature of this chain. Different amplifiers, speakers, and other gadgetry can replace everything except the highly specific digital information on the CD. Surely, then they conclude that this is the cause of my crying. Of course, we know better. We would say that the cause of my crying include Schubert, because he wrote the music, the piano trio, because they played it with such heart-strung inspiration, and the beautiful context in which I first heard the music and first cried as a result of it. This would say, is in my memory and forms the emotional context. We would say that the digital information on the CD is just a way of capturing the moment as accurately as possible and making it possible for me to recreate, partially at least, the original moment. We know also that the information could be coded in many different ways, including analog encoding in the form of a vinyl disc. It's just a database that enables the music to be stored and recreated. In short, we'd have no difficulty at all in laughing at the stupidity of our sailman visitor from another planet. They saw a simple explanation, we'd say, and gabbled at it. How stupid! Well, we should be careful whom we laugh at, for we, too, get trapped in simplistic explanations. DNA mania. Indeed, there is a popular dogma that's reinforced daily in the media, and it must be said, by many scientists, that rests on a crude mistake, just like the Seaman's. Andrea Pichot has called it DNA mania. It's a delusion that DNA could cause life in much the same way as the CD caused my experience of the Schubert piano trio. The analogy is obvious. The human genome is in some ways a little like a CD. It carries digital information. Let's quickly summarize how the genome is all the chromosomes in a cell. A chromosome is a long DNA molecule and some associated protein. It's conventionally divided into genes. A gene is a section of DNA that is used in producing a particular protein. DNA is composed of four chemicals, nucleotides, generally referred to by the letter A, T, G, and C. There are two strands of DNA in each chromosome, wrapped around each other in a double helix. It was discovered of this double helix structure that formed Watson and Crick Nobel Prize winning work in 1953. The nucleotides in one strand always lie opposite in the other according to the rule. A goes opposite T, T opposite C. Two such complementary nucleotides make a base pair. The genome is 3 billion pair base long. These form 20,000 to 30,000 genes. In each gene, the chemicals are arranged in specific ways to facilitate the production of specific proteins. Every time a protein is needed, the appropriate chemical code is read off the gene. This gives the pattern of chemical element that will make that protein what it is. Our genes encode the sequence of the 100,000 or so protein that make up the human body. No protein is made that it's not coded for a gene. So the genome is important. After all, proteins are crucial for life. A living cell is a continuous action-packed drama. Molecular interact and change. One change triggers another, and so on and on. Complex chain of molecular interaction happen again and again. We call them pathways. There are cell cycle pathways, which correspond to the cell turning over. There are development pathways, because cells grow, divide, and form more cells. There are also sort of regulatory pathways, and proteins form the backbone of all these biochemical pathways. Cells organize into tissue, such as skin, bone, muscles, to form organs such as the heart and kidneys. And finally, all this together with the immune and the hormonal system from the organism, the whole animal. This operates in many different ways at various levels of organization, and all of these functions, as biologists say, involve proteins. The causality seems to be entirely one way. The DNA codes a protein, the protein codes a cell, and so on. The organism itself is just what shows on the outside. What really happened, the inside story is that the information coded in the gene is being expressed. In some biology speak, the phenotype is created by the genome, the genotype. The story is addictive. We have fitted ourselves out with a magnificent set of blinders. We have rendered ourselves incapable of looking at the relationship between the genetic code and the living system in any other way. This chapter asks why. Why are we so fond of gene-centered view? We can explore this question by examining a classical and very popular statement of this view. Broken 1976 description of the self-gene. How did so many people come to interpret this view as genetic determinism? That question is particularly important since, as I will show, it's not that of Dawkins himself. Let us, when explore the historical context out of which DNA mania has developed. We start with the reductionist causal chain. This is the inside story that we have just discussed. Skeptically, it looks like this. There is a diagram called the reductionist causal chain. Organism, so I'm going to start from the lower part. The gene codes the protein. The protein codes the pathways. The pathway codes the subcellular mechanism. The subcellular mechanism codes the cells. The cells cause the tissues. The tissues cause the organs, and finally the organs cause the organism. The chain pulls upward. It's a one-way system, from the gene to the organism. The idea is that if you knew all about the lowest level element, gene and protein, then everything about the organism would be clear to us. We could work out what happened at the higher levels and explain it completely in terms of our lower level knowledge. We could reconstruct the whole organism from the bottom up. The first step in the chain is fainter than the other because it represents a rather different cause relationship. At each stage above this one, we are talking about physical cause, how one chemical reaction leads to another. But at the first stage, something different is happening, over and above the physicalization of the chemical reaction involved. It's generally described as the reading of a code. There is transcription and translation of the code. This code is sometimes called the blueprint of life or the program of life. Following Monod Jacobi qualified idea of le programme génétique. I don't know how to read French. So much for the diagram. The problem with it is that it shows only half the story. When we get to chapter 4, we shall see how much it misses out. But for the moment, let us assume it is as comprehensive as it's supposed to be. On that assumption, then let us ask, does the causal mechanism work in the way that we represent here? By no means. Problem with the genetic determinism. Genes are called a DNA sequence. It's this sequence that are replicated and passed on to future generations. So biologists also call gene replicator. Gene determinism somehow sees them as causal agent. How can that be? After all, what does DNA do? As biological molecules go on, Not much. The real player in the action of life are the protein. They are the really active molecules. They indulge most in the biological process necessary for life to occur. DNA is in comparison rather passive. Protein are produced in tiny factories inside the cell of the body. Biologists call them ribosomes. These factories get going when they receive a message that tells them to make a certain protein. Each such message is generated using DNA. A DNA sequence that corresponds to the relevant protein sequence is copied onto another molecule appropriately called messenger, which transmits a form of the sequence to the ribosomes. The messenger molecule, called the messenger RNA, ribonucleic acid, are another type of nucleic acid sequence. The DNA sequences are therefore a kind of template, a specific sequence of nucleotide that can be transcribed to produce the message that is then translated into an amino acid sequence when the protein is made. Amino acids are the unit of which protein is composed, just as nucleotides are the unit of which DNA is composed. That process is called gene expression. This terminology gives the impression that the whole process is implicit in the gene, or at least in the information that the gene holds, which simply needs to be expressed. But it's a little odd to say, as we often do, that the DNA sequence determines the protein. In fact, the DNA just sits there, and occasionally the cell reads off from its sequence and that it needs. In order to get some protein produced, this looks very much like hi-fi equipment reading the digital information on a CD to generate the real action, the music. So the first step in the reductionist chain of cause and effect is not a simple cause-effect event at all. When a sequence is read off, that is an important event, which initiates a whole series of subsequent events. These are physical events too, but it is the process of reading that message as well the object that is read. This process involves certain system of protein. If you wish to identify an agent of the action, it must be those system. They read the DNA code. DNA does not think outside the context of a cell containing this protein system, just as the CD can do nothing without the CD reader. So, you have the paradox that proteins are required for the machinery to read the code to produce the protein. I will return to this paradox in later chapters. But is it just a technicality? Whether we start a causal chain from gene or protein does not matter much, perhaps. Don't we just have to adjust our story slightly and say that the genetic code lies in the protein sequence? That might be a reasonable way of looking at the matter, except that it assumes that each gene is coded directly for a single protein. That is, that the two sequences, the DNA and the protein, are straightforward identical, but they are not. In higher animals, the bits of DNA code that we lump together and call collectively a gene are not always continuous. In man, perhaps most cases, they are broken up into segments. Those segments, called exons, are separated by no-code thread dominantly called introns. The exons code can be combined with various others to produce a full protein code. In ways we do not yet understand, the DNA threads are folded in a three-dimensional form in the nucleus of, of each cell. They cannot exist as a straight thread since each cell contains 2 meters of DNA, which is around 100,000 times longer than most cells. The way the threads are folded inside the cell may make the reading of certain sequences easier than others. There can therefore be many different ways of reading the separate exons and join them up. Technically, there are often many splice variants of a gene, which can therefore code for a set of different proteins. These splice variants are the different ways of reading the separate exons and join them up. Thus, if a gene consists of three exons, it could be read as the forms A, B, C, A, B, B, C, A, C, A, B, C, and perhaps even C, B, A, C, A, B, A, each of which would encode for a different protein. At present, we do not know the rule for which combinations are possible and the use encoding for protein. Consider the gene called the DASCA in the fruit fly Drosophila. It has 110 introns and therefore 10 of 1,000 possible splice variants. Moreover, the DASCA gene does not always operate in the same way. It changes its role with the life cycle of the Drosophila. At any one stage, some of the theoretically possible splice variants work and others will not. At earlier and later stages, the picture will be different. To an extent, it depends on cellular environment. For instance, if there are proteins that affect the transcription of DNA sequence, some activate transcription, some inhibit it. They interact in a complex way. At the same time, there are features in the DNA code itself that influence whether a particular variant can be expressed. Within the DNA sequence of a given gene, we find promoter element and the enhancer sequence. So the regulation of gene expression, as you say, involves a multiplicity of factors which operate and interoperate in several ways. There is regulation of how the code is read off from the gene in order to form the protein transcription. And the regulation of happen after transcription. These are all complicated processes subject to many other influence than the DNA code itself. What this means is that there are many different ways to read a genome. So my analogy with a CD is limited. When you put a CD into your hi-fi, there's only one way you can go on to get music out of each track. This process involves a single one-way readout. When it comes out to gene, by contrast, you have flexible combinatorial readout. A clever CD reader can always do this some extent. We can program our hi-fi equipment to say the music tracks in flexible order depending on how the record has been broken up to form individual tracks. The difference is that the genome is broken up to any unimaginable greater degree, the consequence of which we explore in Chapter 2. Including this flexibility are many backup process. So it's possible to correct for errors and failures at genome level. Indeed, it can happen that an important gene is completely knocked out, and the organism will still manage to get by. If plan A does not work, plan B kicks in. The cell is still able to form protein which function in place of the no-function gene was originally used for. To this multiple influence at the bottom level of protein production, we must add an important higher level complexity. This is that there is no one-to-one correspondence between gene and biological function. Strictly speaking, therefore, to speak of a gene as the gene for A is always incorrect. Main gene product, the protein, must act together to generate biological function at a high level. If you must use the expression gene for X, then you should at least add the plural and speak of genes for X. If this way of speaking is, however, seriously misleading, not only do many genes cooperate in coding for the protein and they interact to produce any given biological function, each gene may also play a role in many different functions, which makes it difficult to label gene functions. I'm speaking here about a higher level function organism. Let's think some of these functions. The pacemaker rhythm of the heart would be one. Another would be the secretion of insulin by the pancreas. And then we could take the transmission of impulse in the brain. Then let's think of the lower level biological process involved in those functions. For instance, there is the process whereby calcium ions get pushed out the cell. Certain identifiable proteins combine to produce this effect. They are important because calcium is used as a control of many processes in cell and organism. This process of moving cation ions goes on in all sorts of way and contexts. For instance, it's involved in all three of the functions we have identified, and many more besides. In fact, it's hard to think of a single high-level function that does not involve this cation pushing protein. And so, implicitly, the gene that we code for them, I could repeat the same story for many other processes in cells. Many of the lower level processes are used again and again in many different functions. High level functions are therefore like a game of recombinations. Suppose you sat down to identify the roles that those genes play in high level function. We'd end up with a list of that we end on almost forever. That's what happens when you start to study how biological function emerges. We get involved in almost endless games of recombinations. So why it is relatively easy to label gene with lower level function, which protein they code for, it's much more difficult to label gene with a higher level function. We need a manual that lists all the functions a gene is involved in and how it contributes to it. Nature does not provide one. We have to work this stuff out for ourselves. That is the research project we call genome ontology. And to get anywhere with that project, we have to look beyond gene and protein. We need to study higher level functions. This is my primary reason for opposing the otherwise colorful metaphor described the genome as Book of Life, Chapter 3. A book may describe, explain, illustrate, and may do many other things, but if you open it up to find just strings of number like the machine code of a computer program, we would surely ask what is the book itself. We would say that we had only been given a database. We could perhaps use another interpretation program to generate a book from each, but until then, all you would have would be a mess of ciphers. My central argument will be that the book of life is life itself. It cannot be reduced to just one of the database. For let's be clear that the genome is only one of the database. Functions in biological systems depends also on important property of matter that are not specified by genes. We will turn to this on chapter 3. Hey guys, I will stop here. So I'll stop here, and if you have let's say any question, just feel free. I guess that's it. I will. So that's all for today. If you have any question, feel free to ask.